Welcome to this targeted oncology presentation in precision medicine called TRK inhibitors, a tumor-agnostic approach to the treatment of solid tumors. I'm Dr. David Hong, Deputy Director of the Department of Investigational Therapeutics, Associate Vice President of Clinical Research at MD Anderson Cancer Center in Houston, Texas. Targeted therapies continues to move towards the forefront of treatment of patients with cancer, and today we're going to talk about an important new therapeutic class called TRK inhibitors that has shown great promise among several advanced malignancies, including thyroid tumors and sarcomas. These agents represent a paradigm-changing advance in precision oncology and drug development for rare mutation-driven cancers. Please welcome my colleague, Dr. Marcia Brose, Director for Rare Cancers and Personalized Therapy at the University of Pennsylvania School of Medicine, Philadelphia, Pennsylvania. And joining us later in the series is Dr. Corey Langer, Associate Professor at the Hospital of University of Pennsylvania, Perelman Cancer for Advanced Medicine, University of Pennsylvania. Let's get started.
Can you please tell us a little bit more about TRK and why is it an attractive target for therapy?
So NTRK is a very unique target. NTRK is a family of genes, NTRK 1, 2, and 3, that encodes these family receptors, TRK A, B, and C. Initially, these receptors were identified in the 1980s and 90s to be really key for neuro signaling. For example, pain, weight, temperature regulation, proprioception, etc. But later on, particularly those researchers in oncology identified that there were unique translocations or fusions of these NTRK genes with other partners that led to constitutive or continued signaling in cancer cells, truly driving their growth and their metastases.
Can you tell me what variants of TRK fusions are found in solid tumors overall and what frequency?
That's a really good question. I don't think at this time we know all of the variants of NTRK, and we've discovered many different variants. The most common variant is ETV6 NTRK3, and it is identified across different histologies. There's not necessarily one specific NTRK fusion gene that is identified in one specific tumor type, but we do see ETV6 NTRK3 being the most common across different histologies.
So are there other NTRK mutations or amplifications that are important?
So to date, we do not, NTRK fusion, mutations have really not been identified as key drivers of these NTRK pathway. And also amplifications, it's unknown whether or not they are significant drivers of the pathway. In the phase 1 trial that we conducted, we did enroll a number of NTRK mutation patients, since we did not know whether or not these patients would actually benefit or not at that time. And none of these patients that we did enroll in the phase 1 actually had any benefit.
So how common is it for people with NTRK fusions to have multiple targetable mutations?
No. So overall, we think that the prevalence of NTRK fusions exists in approximately about 1% of all histologies. There are certain types of tumors, such as mass tumors, mammillary analogs, secretory carcinomas of the head and neck, certain pediatric tumors, such as infantile fibrosarcomas, that we think it's almost pathognomonic, almost 100% likely that they will have these fusions. What's interesting is that in more common tumors like lung and colorectal, although the prevalence is very low, 1% or lower, the patients who have no mutations other than NTRK fusions seem to be the ones who probably harbor these fusions. There are, however, a subset of tumors, MSI-high tumors, particularly MSI-high colorectal cancers, that seem to also have a higher prevalence of NTRK fusions.